Hi everyone, I'm Poojak Patel. I'm a senior at IMSA. So today I'm going to be talking about some research I've been doing on Alzheimer's disease, specifically looking at this enzyme called nNOS and whether blocking it can actually protect brain cells. I did this work at Northwestern in the Klein lab with my mentor, Dr. Noir. Alright, let's jump in. Quick overview. Here's what we're going to cover. Background on Alzheimer's, what we did in the lab, what we found, and then this computational model we built to speed things up. So, Alzheimer's is brutal. It affects 55 million people worldwide, and honestly, we don't have a cure. Current drugs basically just manage symptoms. They actually don't stop the disease and can't prevent it from getting worse. The disease is caused by these toxic protein clumps called amyloid beta oligomers, which just destroy neurons. Now, when these proteins attack brain cells, they trigger this enzyme called nNOS, which stands for neuronal nitric oxide synthase, which goes into overdrive. And when nNOS is overactive, it makes way too much nitric oxide, which causes oxidative stress and eventually kills the neuron. So the question I want to answer was, can we protect neurons by selectively blocking nNOS? So here's what I set out to do. My main goal was to see if blocking nNOS could actually rescue neurons when they're being attacked by these toxic amyloid proteins. The hypothesis was pretty straightforward. If we can selectively block nNOS, we should be able to reduce the damage and keep more cells alive. I took two approaches here. First, the classic wet lab, actually treating the cells with the drug and measuring if they survive. But then I also did something a bit different. I built a computational model to predict what doses would work best. The idea was that if we can predict optimal doses computationally, we don't need to waste time and money testing every single concentration in the lab. So that was the innovation here, combining traditional experiments with computational predictions. Let me give you some quick background. So these amyloid beta oligomers, we call them ABOs for short, they're basically small clumps of misfolded proteins. Unlike the big plaques everyone talks about, these small oligomers are actually way more toxic. They're what actually kills neurons in the early stages of Alzheimer's. Now, nNOS is actually a normally good enzyme. It makes nitric oxide, which neurons use for signaling. But when ABOs show up, they make nNOS overproduce and produce too much nitric oxide. So that leads to oxidative stress, which is basically like rust forming inside the cells. And that's what kills them. So the idea is, if we block nNOS, we break that toxic cycle. This diagram shows what's happening at the molecular level. ABOs bind to receptors, in this case the NMDA receptor, and calcium rushes in, which activates nNOS, and boom, you get this cascade of toxicity leading to cell death. Our nNOS inhibitor, shown here in green, blocks that pathway right at the nNOS step. So even though the ABOs are still there attacking the cell, we're preventing the downstream damage. For the experiments, I mainly used these cells called HT22. They're mouse brain cells from the hippocampus. They're really commonly used in Alzheimer's research because they're super sensitive to oxidative stress, which makes them perfect for testing neuroprotective drugs. I also used MT65 cells to validate everything. These are human cells that actually produce amyloid beta on their own, so they mimic Alzheimer's pathology pretty well. The cool thing is I'm testing in both mouse and human cells, so if it works in both, that's a much stronger case if it was just like one cell line. And a quick rundown of what I used. So both those cell lines, amyloid proteins, an nNOS inhibitor from the Silverman lab at Northwestern, and this assay called cell titer glow that basically measures how many cells are alive by detecting ATP levels. So for the methods, I made amyloid beta oligomers from scratch. You take synthetic peptides and get them to clump under specific conditions. Then I treated HT22 cells with 200 nanomolar of these oligomers for 24 hours. That concentration is toxic enough to kill a bunch of cells, but not so toxic that everything dies. So that allows you to really get like a middle ground to like test if a drug can actually rescue them. Then for the drug part, the nNOS inhibitor came from the Silverman lab, and it's really selective, nearly 100 times more likely to hit nNOS than any other related enzymes. That's super important because if you block the wrong enzyme, you could cause other side effects. So I tested four different concentrations: 12, 25, 50, and 100 micromolars. And the goal was to find the sweet spot where you like get the maximum rescue before you start seeing too much toxicity from too much drug. So the results. First I had to make sure the model and everything worked. So as you can see from the bar graph, treating cells with ABOs dropped viability by about 33%. And you can also see that in the microscopic images on the left, the cells look way more stressed and almost rounded up after the ABO treatment. So this really confirms our baseline, and we really have a working model of toxicity that we can now try to rescue. So here's what it gets more exciting. So the nNOS inhibitor did actually work. So at lower doses, like 12 micromolar, we saw some rescue, but nothing too much. At 25 micromolar, things got a little bit better, but at 50 micromolar, out of these four dosages, that's our more optimal one, as we got massive rescue. And we're talking like viability that goes back to like closer to the actual levels. But the interesting part was that at 100 micromolar, the viability dropped again. So this is actually good news because it means that we're not just seeing specific drug action, or we're seeing specific drug action, not anything just like random. So like too much drug, you're really like starting to get off like you're really starting to attract like toxicity. So this is also where we'll be like looking into afterwards, where it's like more of like a upside down parabola where you see like there's a peak dosage where like too much or too little of it can lead to like over toxicity. Okay, so here's just a graphical representation of this of the cell titer glow assay and just H222 cells for like visualization. Um, and yeah, just now the like validation in MC65 cells. So like obviously like one experiment in cell type wasn't enough, so I repeated them in MC65 cells, and I basically got the same results. So this is huge because it means that isn't just a mouse thing, right? The nNOS pathway seems to work the same way in human neurons. So yeah. To sum up the lab work, ABOs definitely kill neurons, and nNOS inhibition definitely rescues them in a dose-dependent way. And we've also identified this like therapeutic window. But why does this really matter? Because most Alzheimer's drugs just treat symptoms. This could actually be disease-modifying. It's targeting the underlying toxic processes. And unlike those antibody drugs that need like a ton of money to take, a small like molecule pill would be way cheaper. So why build a model, you might be asking. So now, like, let's go into the computational side. Drug discovery is super slow. So if you really wanted to test like a bunch of compounds at multiple dosages, you can, but you're talking about like several hundreds, up to even thousands of experiments, and that takes several months to do. So the idea here was to just build a model that could learn from the previous data and also like predict like the date the predict other dosages, like based on other dosages, the like cons- the viability. So what I did was I used polynomial regression, which is basically fitting a curve into the data, and specifically, degree two. It's a quadratic equation, right? And I trained it based on all the previous data. So, yeah. Um, so we're really just making sure that the model doesn't like memorize the training data and actually like learns the pattern. So for this, I used something called ridge regularization. Um, moving onwards, we also like saw like a inverted U shape, right? So once it's like fully trained, you can really like see like what happens at like 30 micromolars, for example, and it'll give you like an estimation without you needing to like run a specific experiment at that dose. With the which is what we're going to go into now. So the model did perform really well. There was an R squared of 0.84, so like 84% of the variance explains 84% of the variance in the data, right? So like for these kinds of experiments, anything above like 0.75 is considered really good. The average prediction error was around 14%, and the model predicted that the optimal dose would be 45 micromolar, which is pretty accurate to like our own results. So it's only 10% off like from before, but it really just shows how like how much potential like predictive drug development has. So getting a good R squared doesn't unnecessarily mean that the model is legit, right? So you really need to check for overfitting. So I looked at the residuals. Yeah, I looked at the residuals, and which are the prediction errors, and they're pretty small. In fact, the biggest error was only about 1500 RLU, so not too big, and they're randomly scattered. So there wasn't any like pattern. So if the model was overfitting, you'd see like some doses perfectly, but completely miss others. But this wasn't like that. So we're like this is a very good baseline for like being able to screen new compounds. So the impact of this project, right? So like what does this really mean? So for traditional screening, let's say like you're testing 100 compounds at 10 doses each. That's 1,000 experiments, nearly 6 months to a year worth of work. With the model, you test maybe 4 doses per compound, fit the curve, and let the model predict the rest. So now you're down from like to only 400 experiments over like 2 to 4 months. So that's significant like time reduction and like that burden that you have to go through. Limitations and future work. So let's be real about the limitations because every study actually has them. Like first, the limited data, right? So 5 doses from one compound. This is actually this project was meant to be a proof of concept, especially just like the model. So like it's not fully validated, but it works is what it's supposed to show. So what's next is we're going to test the model on more compounds to see if the predictions like still manage to hold up, or even like create a more like in-depth model that's not necessarily just a inverted U curve. That's typically like the default for therapeutics, but we'll see if we can complicate that. So just like to wrap up, ABOs kill neurons, and nNOS inhibition was able to rescue them, and we've also identified a therapeutic window. And on top of that, we've also built a computational model that predicts optimal doses with an 84% accuracy and also helps cut screening time. So this is, it shows how much computational tools have a place in drug discovery and to make it more efficient. So like you don't need to brute force every experiment, and just like you can be more strategic and let computation guide you through these types of processes. So like clinical implications. There's significant potential for therapeutic development, and like this really, from a research angle, this would really like be opening up new questions about the nNOS pathway and even like other pathways for other compounds and how they interact with like tau pathology, for instance. And yeah, overall like computational tools have like a very wide use case in not just neuroscience, but like Alzheimer's, any sort of like other field as well. So. And lastly, I'd like to finish up with acknowledgements. So before I finish, I just wanted to acknowledge the Klein Lab, the lab I did this like research project with, and especially my direct mentor, Dr. Agad Nawar, who guided me throughout the project, and also the lab which I got other compounds from, the Silverman lab, and also my school team, the SIRT.